Hey guys, are you ready for topic 3.3 meiosis? The essential idea of today's lecture is that alleles segregate during meiosis, allowing new combinations to be formed by the fusion of gametes. So let's start by reviewing for a minute exactly what we're talking about, because we're jumping back into topic 3, which focuses on genes and chromosomes and genetics. So this is a picture of a human karyotype. We talked about these previously. Remember, humans have 23 pairs of chromosomes. We have autosomes, which are pairs 1 through 22, and autosomes are any chromosome that's not a sex chromosome. And then our 23rd pair are our sex chromosomes, X and Y. If you are XX, you present as female, XY, male, except for those kind of abnormal cases that we talked about before. And just so that we're clear, you have two sets of chromosomes, right? You have two in each pair because you get one set from your dad and one set from your mom, right? And when you have two sets of chromosomes, we call that diploid, or 2N. Now, most of the cells in your body are diploid, right? Anything from brain tissue to muscle tissue to skin, all that stuff, 2N. The only cells that are haploid, meaning they have one set of chromosomes, are your gametes, and those are eggs and sperm. Today we're going to talk about meiosis, which is basically how we get from a cell that is diploid, right, to a haploid cell. That's the process of meiosis, the process by which both sperm and eggs are formed. So let's review some of the stuff we already know and talk about some new terms that are going to be useful in this lecture. So first, you see a chromosome, right? That's one chromatid. It's one long skinny thing. We've got the P arm, which is up top. We have the Q arm, which is on the bottom. And then we have the centromere. Now, during S phase of interphase, this chromosome that's made up of one chromatid is going to duplicate, right? So now we have two P arms, two Q arms, and we still have our centromere. Now, this duplicated chromosome is called a bivalent, and it's made up of two identical sister chromatids. Now, it's still one chromosome, right? It's duplicated itself and it's stuck together. So it's two sister chromatids, right? But it's only one chromosome. So here we have a nucleus with two chromosomes. We have chromosome number 5 from dad, and we have chromosome number 5 from mom. Now, these two together are called homologous chromosomes, meaning they are the same chromosome. They're chromosome number 5. One came from mom and one came from dad. After DNA replication during S phase of interphase, right, they look like this. So now the chromosome from dad, number 5, is a bivalent. And the same thing with chromosome number 5 from mom. It's a bivalent. Now, we still only have two chromosomes, right? But they're in the bivalent form, meaning they've been duplicated. Together, they're still homologous chromosomes, right? Your blue chromosome is homologous to your purpley red chromosome. When you have two bivalents next to each other like this, of homologous chromosomes, we call that a tetrad. These are just some terms to help us get familiar with some of the stuff you're going to learn today. Okay, let's get into our understandings. Understanding number 1. One diploid nucleus divides by meiosis to produce 4 haploid nuclei. So meiosis is the process of producing 4 haploid nuclei from a single diploid cell. And remember, N is our sets of chromosomes. So diploid cells have 2 sets of chromosomes, one from each parent, and haploid cells have a single set of chromosomes. So here we have a nucleus with 2 chromosomes in it, and it's diploid 2N, meaning there's 2 copies of each chromosome. There's one from mom and one from dad. During S phase, our chromosomes duplicate, right? Now we still only have 2 chromosomes, so we're still 2N. Mom's bivalent and dad's bivalent. Together, these are called a tetrad. Now, during meiosis, there's a process called crossing over that we'll talk about in a little bit, where pieces of these chromosomes actually swap. After the first round of meiosis, otherwise known as meiosis 1, right, we become haploid. Now remember, one bivalent is still only one chromosome, right? It doesn't matter if it's 1 chromatid or 2, it's still 1 chromosome. So we have now effectively gone from a diploid cell that's got 2 copies of each chromosome to haploid, 1 chromosome, right? After meiosis 2, we become haploid, reducing our chromosome number by half. We're back to our single chromatid chromosome. Now, that may seem a little overwhelming, but don't worry, we're going to take some time and go through each step. This is just a basic overview of the process. So, why do we need meiosis to begin with? Well, it's a pretty important part of sexual reproduction because it produces haploid cells. Because gametes are haploid, right? They have half the chromosomes, they're able to fuse together to create a new diploid cell. This is why you look like both of your parents instead of just being a clone of one of them, right? So you have sperm and you have egg, you add those together, that process is fertilization, right? That together forms a zygote. Now, through mitosis, that zygote will become either a male or female adult. That's diploid, right? 2N, 2N. Now, through the process of meiosis, that male or that female will produce more egg and sperm, right? Those come together through fertilization to form a new zygote. So without this part, right, this whole cycle wouldn't work. Another way to think of it is this is a reason why you look like all 4 of your grandparents, right? Instead of just looking like one. Or this might be why you look different than your siblings. So here we have 2 germ cells, not germ like sick germ, but germ cells meaning the line of cells that are going to become eggs and sperm, right? So here we have 2 cells, one in mom, right? This is mom's cell, and in mom's cell, she has chromosomes from her mom, grandma, mom's mom, and her dad, mom's dad. And then here's a cell in dad, right? And in dad's cell, he's got chromosomes from his dad and chromosomes from his mom, right? 2N. Now, after meiosis 1, the chromosomes look like this. And you can see now you've got chromosomes, individual chromosomes that have genetic material from more than one person on it. Whereas before, this chromosome only had genetic material from one person. This chromosome only had genetic material from this person, right? After crossing over, you've got chromosomes that have genetic information from more than one person. After meiosis 2, we have 8 different gametes that are all totally different from each other. So even though they came from one cell, right, we have 4 individual gametes that each have a completely different arrangement of genetic material from different people. So if we take now one of these gametes from mom and one of these gametes from dad and put them together, right? Now you have a zygote that's got a completely shuffled genetic distribution from all 4 grandparents. And then through mitosis, Eventually this will grow into a small BB. So this is how you inherited genetic material from all 4 of your grandparents. Now that's, sexual reproduction. We know that. Now in comparison, there's asexual reproduction, and this is a little bit different. This doesn't require 2 parents, right? You have parthenogenesis, which is when a living creature that typically goes through sexual reproduction produces live young on its own. There's binary fission, and that's how bacteria divide. They just kind of like duplicate everything and split in half, right? There's budding, which tends to happen with things like plants. And then there's fragmentation, like in worms, right? Where you like cut one worm in half and it becomes 2 worms. Please don't try that at home. It doesn't work all the time, and it's pretty gross. But yeah, that's fragmentation. I think sea stars do the same thing. You can like cut a piece off and it'll grow, or the part that you cut off will still be alive for a little bit. It's weird. Anyway, no gamete formation, right? And offspring are identical to their parents. And if you're interested in how this works, there's just recently a Komodo dragon at the Chattanooga Zoo, like this February, actually laid some eggs and hatched, and she had not done so in the traditional way. So if you want more information about that, you can look up that news article. Okay, so that's understanding number 2. The halving of the chromosome number allows a sexual life cycle with the fusion of gametes. Okay, understanding number 3. DNA is replicated before meiosis so that all chromosomes consist of 2 sister chromatids. Now, we talked a little bit about this in the introduction, but let's go over it one more time. So, before meiosis begins, chromosomes are replicated during S phase and interphase. Afterwards, each chromosome consists of 2 genetically identical sister chromatids joined at the centromere. So here we have our chromosome from dad. Here we have our chromosome from mom. Together they are called homologous chromosomes, right? They duplicate during S phase of interphase. Now they are bivalent. We have our bivalent from dad, chromosome number 5, and our bivalent from mom, chromosome number 5. Together, these are still homologous chromosomes, and now they form a tetrad. So, by now, you should be able to do a couple of important things. You should be able to define meiosis. You should be able to distinguish between chromatids, chromosomes, bivalents, and tetrads. You should be able to contrast the number of chromosomes in cells at the beginning and the end of meiosis with that of mitosis. And you should be able to identify the 3 major processes and types of cell division required for sexual reproduction. Okay, now we'll talk about the individual stages of meiosis. So as an intro, remember, the process of meiosis is made up of 2 cell divisions that produce haploid cells. We have meiosis 1, which is where we go from diploid to haploid, and it's composed of 4 subphases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Wow, that sounds a lot like mitosis, right? After telophase 1, or telophase 1, we have meiosis 2, where we go through another set of cell division to create 4 haploid cells. Also, prophase 2, metaphase 2, anaphase 2, telophase 2. Now, I know when we talked about mitosis, we said that cytokinesis was its own stage, but for the most part, we consider cytokinesis to be a part of telophase in meiosis. Why? I'm not sure. It's just one of those things. Okay, so let's take this step by step. Prophase 1. Just like in mitosis, we start to see the breakdown of that nuclear envelope. The nucleolus disappears, and our chromosomes begin to condense and become visible under a microscope. One thing that's different about meiosis is that synapsis occurs, and this is when bivalents of homologous chromosomes pair up to form tetrads. You can see here we have a tetrad here, and you can see crossing over start to occur, and we have a tetrad here where crossing over is starting to occur. Crossing over is when sections of DNA can be exchanged between bivalents in a tetrad. After crossing over occurs, our chromosomes fully condense. So there are three major events that you should know that occur during prophase 1. That's your chromosomes condensing, synapsis, and crossing over. This is understanding 4. The early stages of meiosis involve pairing of homologous chromosomes and crossing over, followed by condensation. Crossing over during prophase 1, in which sections of chromosomes are swapped, results in new allele combinations. And we talked about this a little bit already, but just to be clear, right? So here we have homologous chromosomes, right? We have this chromosome here. It's a bivalent, and we have a bivalent here. Here we can see crossing over start to occur. When this process is over, you'll have parts of your chromosome that have actually switched. Now, it's important that exact pieces change because the gene for eye color is right here, and because these are homologous chromosomes, the gene for eye color is going to be over here, right? So you want to make sure that when chromosomes are crossing over that you swap equal parts or you have equivalent exchange going on. Otherwise, you could have this person has multiple genes for eye color and maybe this person doesn't get any for hair color. So that's not going to work out. You got to have equivalent pieces. Places where crossing over occurs are called chiasma, or if you're talking about multiple, it's chiasmata. And that's just the location at which crossing over between homologous pairs occurs, right? So wherever there's crossing over occurring, we call that a chiasma. Okay. Understanding number 5. Orientation of pairs of homologous chromosomes prior to separation is random. So, in metaphase 1, this is after prophase, spindle microtubules attach to the kinetochores, which are anchoring sites on each of the sister chromatids, which are still connected at the centromere. Okay, so here we have a homologous pair of chromosomes, a tetrad, right? Or, well, these together would be a tetrad. But now we're going to start separating our tetrads. So here are our centrioles, because we're talking about animal cells. Here are the microtubules that are attaching. Now, you notice it said attaching to the kinetochores. These little dots are the kinetochores. Don't get them confused with the centromere. The centromere is the place in the middle of a pair of homologous chromosomes where they are attached. So these bivalents are still attached by the centromere, but these little white dots, these are the kinetochores. Each chromatid, right, has its own kinetochore, and these tubules are going to attach to these kinetochores. Now, the bivalents of homologous chromosomes align at the cell's equator. The orientation of the maternal and paternal chromosomes is random, meaning you don't necessarily know which way they're going to go. It doesn't split so that all the maternal go one way and all the paternal go another way. That's not how it works. As long as each new cell gets one copy of each chromosome, that's all that matters. Okay, now remember, we have homologous pairs. So this is now, you can see there was some crossing over. So this chromatid, right, has both maternal and paternal genetic information on it. This whole bivalent is going to come this way. This whole bivalent is going to come this way, right? The proportion of genetic material from each individual is random. Understanding number 6. Separation of pairs of homologous chromosomes in the first division of meiosis halves the chromosome number. Well, that makes sense, right? Because we're trying to go from diploid to haploid. Now remember, you have two sister chromatids here, but this whole thing is still considered one chromosome. So during anaphase 1, spindle fibers contract and separate the bivalents from the tetrad. This halves the chromosome count of each cell from diploid to haploid. In telophase 1, bivalents reach the opposite poles, so we pulled our chromosomes all the way over. That nuclear envelope is going to start to reform around each haploid set of bivalents. That cleavage furrow is going to start forming, and cytokinesis begins to occur, which eventually results in two distinct haploid cells. Now we're going to move on to meiosis 2. Meiosis 2 starts with prophase 2. Whatever portion of that nuclear membrane reformed, it's going to break down again. And the spindle apparatus, those microtubules, are going to start forming again. Our centrioles are going to move to each pole again, right? And those spindle fibers are going to attach to the kinetochores, right, of each of the bivalents. So pretty much just like last time. Metaphase 2 is when chromosomes align along the middle, right? Metaphase, they line up in the middle. Which pole each sister chromatid faces is random. And then we have anaphase 2, where those spindle fibers contract and separate the sister chromatids, so we're breaking those bivalents now at the centromere. And now each chromosome is formed by a single chromatid, right? And they're pulled to the opposite poles. Here we have telophase 2. Those nuclear membranes are going to start to reform around the 4 haploid nuclei. Those cleavage furrows are going to start forming, and eventually you're going to have cytokinesis, which is going to give you 4 haploid cells, either egg or sperm. So, in review, we have meiosis 1, where we go from diploid to haploid. And then we have meiosis 2, where we go from haploid to haploid. Now I know what you're thinking, how can this be haploid if you see 4 here and 2 down here? But remember, this whole X right here, this is one chromosome. It's bivalent, right? So we still have a haploid set of chromosomes, right? And then we pull them apart. We get our chromosomes back down to one chromatid. And we have 4 functional gametes. So let's see it happen, right? We have our centrioles moving to the poles. We have crossing over. We have our formation of our tetrads. They're lining up, pulling apart. Nuclear envelopes reforming. And now we have 2 haploid cells. Now we're going into meiosis 2. Lining up in the middle. Splitting apart. Telekinesis and cytokinesis. And 4 functional gametes. All right, so meiosis 1, prophase. Crossing over and synapsis. Metaphase lining up in the middle. Anaphase, pulling our tetrads. Telekinesis, telophase, prophase 2, metaphase 2, anaphase 2, telekinesis, and cytokinesis. All right, so that was meiosis. I'm going to let it play one more time. Alrighty, ready, and 4 haploid cells. Good. All right, so what you should do now is make a comparison between mitosis and meiosis. I know they seemed pretty similar, but there's actually some pretty significant differences. So what you should do is use page 52 in your textbook and pages 164 and 165 to create your own comparison. And you're going to use this for skill 1, which is drawing diagrams to show the stages of meiosis resulting in the formation of 4 haploid cells. So it should look something like this, right? You use page 52 and 164, 165. You're going to draw and annotate your own comparison of mitosis and meiosis. Make sure you're using 2 whole pages in your sketchbook side by side so you can compare. It should look something sort of like that. Obviously, there's no annotations on there because that would be really small, but you should definitely have annotations in yours. Make sure when you're drawing the processes of mitosis and meiosis, the number of chromosome sets, whether it's N or 2N, changes to chromosomes like crossing over, and the movement of chromosomes should be shown. Now, this means you're going to have to use more than one color because you're going to have to show crossing over. Okay? And I have actually seen this asked on exams before. So you definitely want to do this. This is definitely something that's fair game for a skills test. Okay. Now let's talk about what happens when this process goes right and what happens when this process goes wrong. Now, the good thing is when this process goes right, you end up with a huge amount of diversity. When this process goes wrong, you end up with things like chromosomal abnormalities, and we've already talked about that. So this slide looks familiar, but now we're going to use it for understanding number 7. Crossing over and random orientation promotes genetic variation. So, genetic variation is necessary for species to change and evolve over time. Now, when we think of evolution, we think of mutations, right? And if a mutation is advantageous, it's more likely to spread throughout a population because it gives you some sort of preference or benefit in surviving. Now, you got to remember, it's not just simply a mutation that is advantageous. There's got to be a combination of the new trait and the environment, right? And sometimes it's not a singular trait. Sometimes it's a combination of traits. It's the way that a mutation or a gene is expressed that makes it advantageous or disadvantageous. So when we say you need genetic variation for a species to change and evolve, you need mutations to cause new alleles, new traits, right? But it's not just the presence of a new allele, it's the combination of alleles. And that's where crossing over comes in. Because you can see here, right here, you may have a new combination of traits that's never been seen before that provides a huge advantage in terms of survival or reproduction, right? So it's the combination of traits that makes crossing over so important. And then there's just the sheer numbers of variability, right? So this is that same slide that we looked at earlier. We have that one germ cell in mom that through oogenesis is going to produce 4 eggs. Yeah, I did say oogenesis. That's a thing. It's the production of eggs, right? And here we have that one cell in dad that's going to produce 4 different sperm. Now, there are 16 combinations of these gametes, right? So just from these 2 cells alone, there's 16 different possible zygote formations that could happen. And that's just 2 cells. You got to remember, mom produces 1 to 2 eggs every month from puberty all the way through menopause, and dad is producing sperm once he hits puberty for the rest of his life, right? So lots of different combinations there. Now, this is also just considering 2 different chromosomes. Remember, humans have 23 pairs, so you can imagine how big that variation is. Random orientation during metaphase 1 and 2 results in a nearly infinite possible of combinations of chromosomes. And there's a little math behind this, right? So if G equals the number of genes in a genome with different alleles, right? Like the allele for eyes, you have blue, brown, or different alleles for hair color, right? Blonde, brunette, really, really dark, all sorts of stuff. 2 to the G power is the number of combinations of alleles generated via meiosis. So let's do just a little bit of math, right? The human genome comprises about 25,000 genes. So if we were to use that math, that's 2 to the 25,000th power, right? 5.622 times 10 to the 7,525th power possible combinations of alleles. That's insane. Remember, there's only like almost 8 billion people on the planet. We're nowhere near the total possible combinations of alleles. Now remember, not all those combinations is going to be advantageous, or some of them might not make any difference at all. Think about those silent mutations that we talked about. But even still, that's a huge number. Understanding number 8. The fusion of gametes from different parents promotes genetic variation. So, you already know this. Sperm and ovum cells are the haploid products of meiosis in humans, and it's the fusion of egg and sperm, randomly chosen, which further promotes genetic variation of the resulting offspring. So here we have a haploid egg, we have a haploid sperm, right? We have N, put them together, you get 2N, brand new individual, unique combination of traits. Application number 1. Non-disjunction can cause Down syndrome and other chromosome abnormalities. So we talked a little bit about this before. Non-disjunction occurs in meiosis when chromosomes fail to separate correctly. This results in daughter cells with either too many or too few chromosomes, right? So here we have meiosis 1, you have non-disjunction happening. So instead of this tetrad splitting and one bivalent going this way and one bivalent going this way, you end up with 2 cells in meiosis 2 where you have, here's one of the chromosomes, right? And you're totally missing that chromosome over here. This one, on the other hand, got both bivalents. So now we have 2 copies of whatever chromosome number that is, right? So we have extra chromosomes over here and not enough chromosomes over here. So what that means is if you're thinking about those karyotypes that we did, right? This would be, if I were writing the notation, it would be plus an extra chromosome, right? And this would be minus a chromosome. And you can see the same thing happening over here. It doesn't always happen in meiosis 1. Sometimes it can happen in meiosis 2. Now, if it happens in meiosis 2, you only end up with 1 cell that's got the extra chromosome and 1 cell that's missing one. So, the formation of any of these gametes with any of these gametes is going to result in either an extra chromosome or a deletion. We talked about Down syndrome. Down syndrome is a extra chromosome 21, right? Trisomy 21. Yep, there it is, trisomy 21. So Down syndrome is caused by non-disjunction when a zygote receives an extra copy of chromosome 21. And we use a karyogram to determine this. This should sound familiar. I kind of jumped the gun when we were talking about chromosomes and started talking about this stuff because it's just so cool. Now, a couple other things about Down syndrome. Studies have shown that the age of parents can affect the odds of non-disjunction resulting in trisomy, which is a set of 3 instead of 2 chromosomes. As parents age, the chances of having a child with Down syndrome or some other chromosomal disorder increases. And you can see it really starts picking up in the 30s, right? So we're talking about maternal mother's age. Now, that's not to say that it's not an issue on the paternal side. It can be. We measure maternal age because you're measuring the age of the mother when she has the child, but the actual age of the father can contribute as well. So as as men progress in age, the chances of them contributing a sperm with a chromosomal abnormality also increases. Usually show the graph in maternal age, though, just because, you know who the baby is coming out of, right? And you know what their age is. You don't always know if dad. But actually, it's kind of funny because I remember telling my husband after we were married, if we didn't have kids by the time I was 30, I wasn't doing it. And this is exactly why. And Grace was born when I was 29, so we just made it. But you can even see now, like I'm 38 now, and I wouldn't risk it because just that little increase is, a huge deal. And as you get older, right, it continues to go up and up and up. So, application number 3. Description of methods used to obtain cells for karyotype analysis, things like chorionic villi sampling and amniocentesis. So, occasionally, if you don't get genetic counseling done before you have a child, the doctor can tell using sonograms if there's an issue with the child. So there are two methods that we use, two medical procedures that we use to determine if there are chromosomal abnormalities in a fetus after conception. So the first one is amniocentesis, and this is performed usually later in a pregnancy, where you take a needle and you actually take some of the amniotic fluid, and that fluid has cells from the baby in it, and then you can do a karyotype on those cells. The second is chorionic villus sampling, where they insert a catheter and they take a tissue sample from the actual placenta. Now, I don't know if you remember from freshman bio what the placenta is, right? So here's the fetus. You can tell this fetus is much earlier. This particular process is happening much earlier in a pregnancy than amniocentesis because the baby's not nearly as developed. But placenta, okay. So here's the baby, right? There's the umbilical cord. The umbilical cord doesn't just plug into mom. It plugs into the placenta. The placenta is a temporary organ. It's the only human temporary organ that grows. And it grows only during pregnancy. And this placenta is the interface between mom and the baby. So remember, baby has its own immune system, right? Now it's getting antibodies from mom to help it fight disease if necessary, but it's not like mom's immune system is in here running the game. Now, especially if they have two different blood types, that's an issue. There's got to be some sort of barrier between mom and the baby. So, mom delivers oxygen and nutrients and carbohydrates like glucose and things through the placenta, and it's fed through diffusion into the umbilical cord and then into the baby. Likewise, the baby passes materials back to mom, things like carbon dioxide, because respiration is going on. Now, the baby's not breathing, right? But the baby does need oxygen. It is using cellular respiration to use ATP. So it's got to get rid of that carbon dioxide. That carbon dioxide, through diffusion, passes into the placenta, where it's picked up by mom's circulatory system and then through her mom's circulatory system to her lungs and then out. So you can think of the placenta as like the interchange between mom and baby. It helps keep them separate so that baby is isolated, right? But it allows the movement of materials in between. So in chorionic villi sampling, what you're doing is actually taking a piece of this tissue, right? Not the baby itself and not the amniotic fluid. And then you can use a karyotype to look at genetic information from the baby, because there is material coming from the baby in here. Okay, so in amniocentesis, a needle is passed through the abdomen into the amniotic sac. Amniotic fluid is withdrawn, and that contains fetal cells. That fluid in here contains fetal cells. You can see they're using a sonogram, right? This procedure can be done later in pregnancy, but the risk of miscarriage is low to moderate. And I'll tell you my own story with this. When I was about 5 months pregnant, because it was December, I had a sonogram, and I could tell when I was looking at the doctor's face that something didn't look right. And they didn't say what was going on. They just said, we're going to schedule you for a 3-dimensional sonogram. Can you come back on this day? And this was like 2011. No, 2010. So 3D sonograms had just come out. They were expensive. They were extra. They said, no, we're going to come in and have you do that. And I said, oh, cool. Okay. So I came back in and they did a 3-dimensional sonogram, and then I could see what they were looking at. They were looking at my daughter's brain. There were big fluid pockets in her brain. And just from I'm not a doctor, but I can tell that that wasn't necessarily right. So when they finally decided to explain what was going on, they told us, she has these pockets in her brain. We're not exactly sure what they are. They could be nothing. They could go away on their own. Sometimes we see them. Sometimes, they just go away on their own. Or the other alternative is she has a chromosomal abnormality and the life expectancy is pretty much like zero. So my choices were I could do nothing and just kind of see what happened, or I could do an amniocentesis. And I actually decided to not do the amniocentesis because if she had the chromosomal abnormality, then it was fatal anyways, and she wasn't going to live, right? If she didn't have the chromosomal abnormality and she was going to be just fine, I didn't want to increase the risk of a miscarriage just to find that out. So, long story short, she was totally fine. She was born. Everything's perfect. There was no chromosomal abnormalities, and it worked out. And then here we have chorionic villus sampling or villi sampling, where it's that same thing I was talking about just a couple of slides ago. You can do this earlier in pregnancy, but the risks are pretty high. So this is usually reserved for like you're pretty sure that there's something serious going on, and like these are used in cases when the mom's life could be at risk, or they're sure that there's something significantly wrong with the fetus pretty early on. All right, so this you can use this information to make decisions about whether to continue with the pregnancy or not. So by now, you should be able to do a couple of different things. You should be able to identify two major sources of genetic variation. You should be able to define non-disjunction. You should be able to state the relationship between parental age and the likelihood of non-disjunction occurring. And you should be able to identify the methods. Oh, that's not bad. That's horrible writing. Identify the methods of obtaining cells for use in producing a karyogram and the associated risks. All right. And thanks so much for paying attention. And just like last time, if you have any feedback, let me know. I think the audio was probably a little better this time, and hopefully you can see the cursor. And thanks. Talk to you soon.